This conference will now be recorded.
Good morning, students. I'm Dr. Divya from TeenEinstein. So this will be the last class for principles of inheritance and variation. So we have already learned different concepts of genetics. Hope you have gone through all those topics once again. So just I'll give a summary. So genetics means that deals with the principles of inheritance and variations, right? So the progeny, how one progeny is resembling their parents or how that resemblance is coming. About that we are studying in genetics. So many biologists, they are contributed in many ways. They have given their contribution in the field of genetics. And Mendel proposed the principles of inheritance, and now it is called as Mendel's law of inheritance. And he made that, or he brought that concept of factors, and now these factors are called as genes. So first, who proposed the concept of genes or factors? Earlier it was called as factors. So that is by Mendel. Mendel was the first person who brought such a great concept of genes or factors. And from that, all these studies, they continued, other scientists continued. And that was the broad concept what he made. Okay, and he conducted several experiments in pea plants to prove his concepts, and he produced several generations of F1 and F2, and from that he made two laws of dominance and law of inheritance, law of segregation, all these things. Then what is the recessive character, what is the dominant character, if it is coming in heterozygous condition, how that generation will appear, and when it is coming in homozygous condition, how the appearance will be. Everything he explained with his experiments. But in biology always you can see exceptional cases, right? That's why every time in biology new inventions are coming, because this kind of variations, inheritance, everything is there. Okay, so all characters, it won't show true dominance, and some characters will show incomplete dominance, and some characters will show codominance. So about all these studies, exactly like Mendel's concept, things won't work out. There are also variations will come, right? And Mendel studied the inheritance of two characters together. First, single characters, then two characters, how it will get inherited together, and how it will independent assortment of those characters will happen, on how it will combine all these things in different ways he studied. Then we learned about Punnett square, how we can write all the possible combinations of a gene, or in a dihybrid cross, how we can write all this. Reginald G. Punnett, he proposed that concept. Then what is the genotype? Genetical expression. Then phenotype means the physical expression, or the physical character which a genotype can express. That is called as a phenotype. All these things we learn. Then genes are located on chromosome. Then there is a how they correlated Mendel's law and this gene or chromosomes and all. There is a segregation and assortment of chromosome during meiosis or cell division. Then Mendel's law, that law, those laws were extended in the form of chromosomal theory of inheritance. And then what and all we learned? Then we learned about genes like sex-linked genes or sex-linked inheritance, male and female. We found that chromosomes, common chromosome, another set of chromosome which is different in male and female. So all these things we learned. So today we are going to learn about then some important concepts which is only for NEET exam. That also I explained and gave. Hope you have notes of all those things. I'll share the questions. Please do practice those questions because it's not easy as you're thinking. If you are having the full concept idea about genetics, you can easily solve and get marks. For board exam and your NEET exam, you can score very good marks from this session. But you should have a complete concept. The concept should be clear. Then it is very easy to solve these questions. Okay, from this chapter itself, you can score very good mark without losing anything. If the concept is clear, means because you will get so many problems from this session. Then mutation. Today we are going to learn about mutation. Okay. So what is that mutation, or what do you mean by mutation? Mutation means generally we have a DNA sequence, right? So mutation means some changes in that. See, one DNA helix, in that helix some changes. So mutation means that is phenomenon that results in the alteration of DNA sequences. And that alteration, if the DNA sequence is changing, definitely the genotype and the phenotype of that organism will change, right? That we have already told that gene, how it will work. It will code for gene, then it will code for particular character. So that character itself, if there is a mutation means changes in that. So if there is a change, definitely that will change the genetic constitution. So the physical appearance, however, if the genetics changing means the physical appearance will definitely change. So that is called as mutation. So that is a phenomenon that results in the alteration of DNA sequences, and that results in the change of genotype and the phenotype of an organism. That will definitely change the genotype and the phenotype of organism. Then that leads to the variation of DNA. Then what and all we need to learn? The chromosomal aberrations. So the genes are located on chromosomes, that we know. So alteration of that chromosome, that results in abnormalities. See, there will be a normal condition for that chromosome, right? If there is a change or some mutation, that may cause chromosomal abnormalities or chromosomal aberrations. Okay. So the genes that not to be located on chromosomes. Genes are present on chromosomes. So if there is a slight change or mutation, like that changes are there, means that will create chromosomal aberrations or chromosomal abnormalities. So the DNA helix runs continuously from one end to another end in each chromatid in a highly supercoiled form. Okay, DNA is in a double helical form. So that is in a highly supercoiled form. It is in that chromatid, in each other chromatid in a highly supercoiled form. Okay, the chromatids are in a highly supercoiled form. So these chromatids, if there is a deletion or insertion, if some part is getting deleted or some part is getting removed from that, or any insertion or gain, or some part is getting inserted to that, definitely that is making a change to that normal form of the DNA, right? If some part is getting deleted or removed from that DNA, or some part is getting added or inserted to the DNA, that makes a change in the normal constitution or normal configuration of the DNA, right? That makes a change in the protein synthesis, and finally the phenotypic and genotypic expression. So that is called as chromosomal aberrations or chromosomal abnormalities. So these genes, there is a change in genes or gene location, everything. So alteration of all these results in the abnormalities of that particular chromosome or chromosomal aberrations. So these chromosomal aberrations are commonly observed in cancer cells. Okay, these chromosomal changes are commonly observed in cancer cells. Cancer cells means what they have? They are losing the control. Generally, we are saying means it is losing the control. There is see, for a cell and cell division and all, there is a control, right? So here in cancer cells means they are losing the control. Okay. So the chromosomal aberrations or chromosomal changes, normal pathway, it is moving from that normal pathway, and the chromosomal aberrations or changes are commonly seen in cancer cells. And mutations also arise due to change in a single base pair of DNA. Okay. See, almost all points, maximum points I've tried to include in this. That's why point wise I'm explaining. So listen carefully, because each and every point is important in this chapter, because you will get so many questions from this part. So mutation. Mutation also arises due to change in a single base pair of DNA. Okay, single base pair. You know the single base pair of DNA. In that also mutations may happen. So that is called as point mutation. Means, what do you mean by point mutation? In a single base pair, there is a change in single base pair, that is called as point mutation. Mutation means change from the normal condition. So that mutation may arise in a single base pair of DNA. In a DNA, a single base pair may change, or change may happen to single base pair. That is called as point mutation. Only to that particular point that mutation is happening or mutation is coming. Okay. So a classical example of such a mutation is sickle cell anemia. So point mutation example is sickle cell anemia. Point mutation. From that word or that term itself, you can write what is point mutation. Only in single base pair, only in that point it is getting changed or it is getting mutation. So example for point mutation is sickle cell anemia. Then deletion and insertion of base pairs of DNA, that causes frame shift mutation. Right? Deletion and insertion. Sometimes there will be deletion of a particular base pair of DNA, or sometimes insertion. That causes frame shift mutation. So what is frame shift mutation? Deletion and insertion. Sometimes deletion and insertion. Some part is getting deleted and another part is getting inserted. That is called as frame shift mutation. So there are many chemical and physical factors that induce mutation. Okay. These chemicals which are inducing that mutation, they are called as mutagens. Okay. So what are mutagens? The chemicals or the chemical and physical factors that induces mutation. They are called as mutagens. And next is UV radiation can cause mutation in organism. UV radiation is a mutagen. So physical factors and chemical factors that create or that induce mutation, they are called as mutagens. And the UV radiation that can also cause mutation in organism. That UV radiation is a mutagen. Okay. Next is pedigree analysis. So in human society, generally there is a concept that that disorders, physical or that health disorders that are getting inherited, right? So this is based on the heritability of some characters or some features in some families. Usually, we will, some kind of diseases are getting inherited from one that family to that. Throughout the family, it is getting inherited in all generations, like that we used to say. So what is that? It is based on the heritability of how that character is getting inherited. That is also one character, that disease or something, that health issues. Such things are getting inherited. So that and all, the heritability of certain characters in families. So how these characters are getting inherited? So after the rediscovery of Mendel's work, people started analyzing this inheritance pattern of these human traits or all these kind of characters in human beings. They started tracing out the how it is getting transferred from one generation to another generation. See, in plants and also if you are if you are conducting one experiment in pea plant and all, we can control. Like pollination, we can control, or that character from male it is passing to female. Like in that case, we can control the pollination or something we can do. In the case of plants, but what we can do in the case of human beings? In human being, we can't control like that. In next one character is passing to next generation, we can't control, right? So it's not possible in the case of human beings. So only thing we can do that from the family history, we can study the inheritance pattern of particular trait, and that from that we can find out one alternative method, right? So such an analysis, like that one analysis in several generations of a family, okay? So analysis of traits in several generations of a family, that is called as pedigree analysis. So what is pedigree analysis? In the case of plants and all, we can study how one trait is getting passed, and we can take control measures like pollination. In that we can do, or some other methods. In the case of plants and all, we can do. But in the case of human, what we can do? We can't control like that, right? If they are getting one baby, how we can control, like one disease should not go to, or one particular character should not go to next generation? We can't control like that in the case of human being. So only thing we can do is from several generations, we can study how one character is getting passed from one generation to next generation. Some characters we can, in successive generation, we can see the character, right? From mother, exactly that character you can see in girl baby or boy baby or something, or from father some character. But some character, it will skip one generation and it will go to next generation. Like that also characters are there. Okay, so an analysis of traits in several of generations of a family, that is called as pedigree analysis. So in the pedigree analysis, the inheritance of a particular trait is represented in the family tree over generations. Okay, how the character is inherited, or for a particular trait, how it is getting inherited, that character is getting inherited, that we can represent in a family tree. Okay. So in human genetics, pedigree study that provides a strong tool which is utilized to trace the inheritance of a specific trait, abnormality or disease. Okay, here comes the importance of pedigree. So how we can utilize that pedigree analysis? If one character, especially in the case of abnormalities or diseases, it is getting passed from one generation to next generation, that flow of that character we can study, or the inheritance of that particular trait we can study in one family. So in human genetics, that pedigree study provides a strong tool which is utilized to trace the inheritance of a specific trait. We can trace the inheritance of a particular character or abnormality or disease. So in the case of disease and such kind of abnormalities, there comes the importance, right? So here in pedigree chart, here you can see this, how we are representing male, female, then sex unspecified. We don't know the sex. The sex is not specified, means we'll mention it like in this way. Then affected individuals in red color we'll mention. This is affected male, this is affected female, this affected individual, but the sex is unspecified. This is mating. If you are connecting one male and female like this, it is mating. See, mating between relatives will mention with double line, mating between relatives. See, parents above here, male and female, these both show the parents, and here you can see the children. These are the children. From here, see, in order of birth, from left, we are mentioning the birth. This first one, this is second child, like that. Next, third one is here, one more is coming with, that is the third birth. Okay. Then, parents with male child affected with disease. These are the parents, and here, parents with male child, and that is infected or affected individual, that is male. Here, circle comes, means it is affected female. Here, this diamond shape comes means that is affected individual, but the sex is unspecified. Okay. Here, like this, we are mentioning means 5 unaffected offsprings are there. 5 unaffected offsprings. If number 6 or 7, it depends upon the number, but like this, inside number we are mentioning means they are unaffected offsprings. Okay. Next. Next is Mendelian disorders. Okay. So generally, this genetic disorders that can be grouped into two categories, Mendelian disorders and chromosomal disorders. Okay. So genetic disorders we can group into two. All genetic disorders we can group into two, Mendelian disorder and chromosomal disorder. First one Mendelian disorder and second two groups are there, Mendelian disorder and chromosomal disorders. So Mendelian disorders are mainly determined by the alteration or mutation in a single gene. Okay. Mendelian disorders are mainly determined by the alteration or mutation in the single gene. Okay. So two types of disorders are there, Mendelian disorder and chromosomal disorder. So first we are going to learn about Mendelian disorder. Mendelian disorders are that is mainly by alteration or change or mutation in the single gene, that is called as Mendelian disorder. In Mendelian disorder, we are talking about the mutation or change of a single gene. So these disorders are transmitted to the offspring on the same line. Okay, it is transmitted to the offspring on the same line, and the pattern of inheritance of such Mendelian disorders that we can trace by the pedigree analysis. So with the help of this pedigree analysis, we can trace that Mendelian disorder or the pattern of that inheritance of Mendelian disorder we can trace with the help of a pedigree analysis. Okay. So mainly the common Mendelian disorders are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia. So these are mainly the Mendelian disorders. Which are all the Mendelian disorders? Hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia. So example for Mendelian, don't get confused, these are Mendelian disorders. Hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, then thalassemia. These are Mendelian disorders. So these type of Mendelian disorders may be dominant type or recessive type. So they may be dominant or recessive type. So by pedigree analysis, we can easily understand that it is dominant or recessive. Okay, that particular character or that particular trait is dominant or recessive. We can make out very easily with the help of this pedigree analysis. And in the case of hemophilia, that may be linked to the sex chromosome also. And X-linked recessive trait shows transmission from carrier female to male progeny. Okay, hemophilia is related to sex chromosome. Which chromosome? Hemophilia is related to the sex chromosome. Okay, so that X-linked recessive trait shows X-linked which chromosome? Sex chromosome. In sex chromosome, that X-linked recessive trait shows transmission from the carrier female to the male progeny. Transmission from where? From the carrier female. That female is a carrier. She is not having that disease. That female doesn't have that disease, but that can act as a carrier to the male progeny. To the male progeny, it will come. Okay. And next, here you can see that pedigree analysis of autosomal dominant trait. Okay, this is the representation of autosomal dominant trait. See, I have explained all this. Here, this is red means what? Red means infected, and here this is round. So that is infected female with normal male mating. This shows mating, and in that all this. These are next generation. From left onwards, we need to check. This is the first one, first child, uninfected. Here, but that is female. Here second one, that is infected male, and here again uninfected. Then the fourth one is here infected female. Okay, and this shows the mating here, normal with normal. This both are not infected, so their progeny without any infection. Okay, but here infected female is getting married or getting mated with uninfected male. Okay, in their progeny, how it comes? Infected, which one? Infected female, right? So that's how autosomal dominant trait. These are autosomes. Autosomal dominant trait. How one autosomal dominant character is getting passed. Okay, so that you can learn from this pedigree chart. So autosomal dominant trait example is myotonic dystrophy. Myotonic dystrophy. So with the help of this, you can easily read this pedigree chart. Okay. And next one, exactly like this, we have given autosomal, that is the dominant one. Here, it is the recessive one. Sickle cell anemia. Sickle cell anemia, that is the recessive one. So here, uninfected, both are getting mated, and here the male. In the progeny, the male is getting infected, and one female is also getting infected, right? Again, so these and all will, how it will be? They are working as carrier, otherwise it won't go to next generation, right? They'll be normal, but that female can work as a carrier. Okay. So autosomal recessive trait, that is, for example, sickle cell anemia. Next, we are going to learn about genetic disorders. In that, first one is hemophilia. First is hemophilia. So in hemophilia, this is sex-linked recessive trait. Sorry, first we will learn about color blindness. Color blindness. So what is that? Color blindness is sex-linked recessive disorder. This is sex-linked recessive disorder, and due to this defect, either red or green cone of eye that will get infected or that will get affected, and it's a failure. In this condition, it's a failure to differentiate or discriminate. We can't make out between red and green color. So that is the color blindness. Blind to those colors. Okay, that is think in that way, blind to those colors. So it is difficult. It will be difficult to differentiate that red and green color because those infected people will be unable to differentiate these two colors. So the disorder due to defect in either red or green cone of eye. Defect in either red or green cone of eye resulting in failure to discriminate between red and green color. They will fail to discriminate between red and green color. So this defect is due to mutation in certain genes. Okay. Mutation of some genes which are present in the X chromosome. So how it is happening? This color blindness, it is due to mutation in certain genes present in the X chromosome. On X chromosome, few genes will be present and those genes will get mutated. So due to that mutation, mutation of some genes which are present on the X chromosome, color blindness is coming. So, in 8% of male, this is happening, and 0.4% of females only getting affected, or females only 4%, 0.4 percentage, not 4 percentage, 0.4 percentage of females only getting infected. But in the case of male, 8%, almost 8% of males are getting this, and female percentage is very less, only 0.4. Okay, so this is because the genes that led to red-green color blindness are on X chromosome. Okay, why in female percentage is less? That red-green color blindness, how it is coming? Those genes are present on X chromosome. So males have only one X chromosome and females have two X chromosomes, right? Females means how will represent? XX. Male means XY. So males have only one X chromosome and female have two X chromosomes. So the son of a woman who carries the gene has 50% chance of a color blindness, right? So the son of a woman's son who is carrying a gene. In the case of male, only one X chromosome will come. So there is a chance of 50% being colorblind. And in mother, that mother is not herself colorblind because the gene is recessive, right? In mother, it will be recessive gene, so there won't be color blindness. But when it comes to male, there is only one X gene, right? So there is a 50% chance in the case of male, okay? But in the case of female, that mother won't be colorblind because there only that recessive gene is present. But when it comes to next generation, that male, they are having only one X chromosome and female have two. Okay, that means that effect is suppressed by matching dominant normal gene, where in the case of female, that effect is suppressed in female by a dominant gene. Okay, but a daughter will never be normally, in normal case, the daughter won't be colorblind because mother is acting as a carrier and father is not. The daughter will not be colorblind unless her mother is a carrier. If mother is not a carrier and her father is a colorblind, unless and until that daughter will never be a colorblind. But in the case of male, there is a chance. Okay, in male 8%, but in female 0.4%. Why? Because in the case of female, there both genes will come, right? Two X genes. There two X genes are there, but in the case of male, there is only one X gene, XY. That is a combination of male. But in the case of female, XX. So there will be a dominant X and recessive X. So dominant X comes. When dominant X comes, that will hide the character of that recessive gene, right? So the dominant X character will get expressed. So the chances are very less in the case of female. Okay, so that is color blindness. Next is hemophilia. Hemophilia means that is sex-linked recessive disease. Okay, this is also recessive disease and that shows its transmission from unaffected carrier female. Here also female who is unaffected and a carrier to some of the male progeny. Okay, this is getting transferred to the male progeny and the female is acting as a carrier and female won't be affected. That unaffected carrier female to some of the male progeny has been widely studied. This, in most of the cases, this is happening. This is a sex-linked recessive trait disease, and that shows transmission from unaffected carrier female to the male progeny, and the female is acting as a carrier. So in this one, a single protein that is a part of the cascade protein involved in the clotting of blood, that is affected. See, there is a group of protein in the blood clotting process. In the clotting of blood, in that process, cascade means several proteins are involved. There is a group of proteins involved in the clotting of blood. But in that, a single protein is involved. Okay, in this disease, a single protein that is a part of cascade protein involved in the clotting blood. Okay, in blood clotting, several proteins are involved. Okay, that is a group activity, or a group of proteins are working to clot the blood. Okay, among that, one protein is getting affected or one protein is getting changed in the case of hemophilia. Okay, so due to that, if a person is affected with this disease, with a single cut, that will lead to non-stop bleeding. So what is the character of that hemophilic patients? Blood clotting. Blood clotting is very difficult, or it is impossible, because group activity, that cascade protein, they are involved. A group of proteins are involved in the process of blood clotting. So in that, among that, one protein is affected, or that one protein is affected due to this disease. So if with a single cut also, there will be non-stop bleeding in such people. Okay. The heterozygous female will act as a carrier, and that will transmit, or that carrier may transmit the disease to sons, only to male. Okay. And the possibility of a female to become hemophilic, it is very difficult, or it is very rare, because in that cases, mother should be a carrier and father should be a hemophilic patient. Okay, so that only in rare cases such things are happening. And in Queen Victoria, in their pedigree, or in their family history, in Queen Victoria's pedigree, a number of hemophilic people or hemophilic infected people were there, and Queen Victoria, she was a carrier. Okay, carrier of this disease, because mostly females will act as a carrier, and in male this disease comes, right? So a heterozygous female for hemophilic, they will transmit disease to son. So that extremely rare cases, only in extremely rare cases, female or the mother, such female has to be, there is a, in female, there is a chance of becoming a infected person. Usually they will pass that to male. Okay. So in the pedigree of Queen Victoria, they were having hemophilic infected people, and Queen Victoria was a carrier of hemophilia. That's why it is called as royal disease. Okay, hemophilia. Next is sickle cell anemia. The next one is sickle cell anemia. So sickle cell anemia, this is autosome linked recessive trait. Okay, autosome. This is not related to sex chromosomes. This is autosome, linked to autosome recessive trait, not dominant trait. Autosome recessive trait, and that can be transmitted from parents to the offspring when both the partners are carrier of a gene. Okay, if both the partners, both male and female, they are carrier of this gene, then they can pass to next generation. This is autosome linked recessive trait, and when both male and female, both parents are carrier of the gene, then they can pass to the offspring. Okay. So this disease is controlled by single pair of alleles, HBA and HBS. Single pair of alleles, HBA and HBS. Okay, so out of these, only homozygous individuals for HBS, homozygous condition. When it comes in homozygous condition, they shows the disease phenotype. Okay. Sickle cell anemia means blood cells will be in the form of sickle. Okay. That is called a sickle cell anemia. So in this homozygous individuals, that means HBS, this homozygous condition, HBS, both should be same. So HBS, both are coming in same condition means they shows the disease. And heterozygous, when it is coming in HBA and HBS, this condition, that in such cases individuals are unaffected, but they are carrier of the disease, and there is 50% chance of transmission of this mutant genes to the progeny. And that's how they exhibit that sickle cell anemia. So this defect, that is mainly caused by the substitution of glutamic acid. Substitution of glutamic acid by valine, where on the 6th position of the beta globin chain of the hemoglobin molecule. So on hemoglobin molecule, beta globin chain is there. In that chain, 6th position, usually glutamic acid should be present. So instead of that, that glutamic acid should be replaced by it will get replaced by valine. So that shows the sickle cell anemia. Okay. So hemoglobin molecule on that beta globin chain, on beta globin chain 6th position, normal condition. In normal condition, glutamic acid will be present. But when sickle cell anemia, this condition comes, that glutamic acid is replaced by valine. So that condition is called as sickle cell anemia. Okay. Here. This is the normal condition. Okay, here you can see. 1, 2, 3, 4, 5, 6. On 6th position, you can see glutamic acid. Right? It's a normal individual. But here, when sickle cell anemia comes, here it is getting replaced by valine. Here on 6th position, you can see. Remaining and all same only. Here, everything is same except here 6th position, instead of glutamic acid, valine is coming. So, that condition shows the sickle cell anemia. Here you can see this is the normal blood cell condition, but when sickle cell anemia comes, it becomes like this. Okay. Next, phenylketonuria. In phenylketonuria, this is inborn error of metabolism. Okay, this creates metabolic error, and it is autosomal recessive trait. It is getting inherited as autosomal recessive trait, and the affected individual, if an individual is getting affected or individual is getting this problem, phenylketonuria, that lacks enzyme that converts the amino acid phenylalanine into tyrosine. All these portions you have to by heart. Okay, there is no other option. You need to note down all these points. So phenylalanine into tyrosine. So that enzyme won't be present in phenylketonuria. It's metabolic error. Phenylketonuria is a metabolic error, and this inherited as an autosomal recessive trait. This is autosomal recessive trait. So the affected individual, they lack enzyme. If an individual is infected or affected with this phenylketonuria, they won't be having the enzyme which is converting the amino acid phenylalanine to tyrosine. That particular person won't be having phenylalanine amino acid, which is that converts the amino acid phenylalanine into tyrosine. Okay. That enzyme won't be present, which is converting phenylalanine into tyrosine. Don't get confused to phenylalanine to tyrosine. That conversion won't take place, or that enzyme which is converting this amino acid phenylalanine into tyrosine, that won't be present. So, if it is not there, this phenylalanine, it will get accumulated. Okay, and that will convert into phenyl pyruvic acid and relatives of derivatives of that phenyl pyruvic acid. Okay, so it will accumulate. Again, this will accumulate, and that will cause the Mendel retardation. Where it will accumulate? This accumulation of this phenyl pyruvic acid and derivatives of that phenyl pyruvic acid, that accumulation in brain, that will cause the Mendel retardation. Okay, so that cause phenylketonuria, and this is also excreted through urine because of its poor absorption by kidney. Okay, so inborn error of metabolism inherited as autosomal recessive trait, and this individual, those individuals, that affected individuals, they won't be having the enzyme which is converting the amino acid phenylalanine to tyrosine. As a result of that, that phenylalanine will accumulate in the brain, and phenylalanine, and that will convert into phenyl pyruvic acid and derivatives of that. So such things, phenyl pyruvic acid and other derivatives, will accumulate in brain, and that cause the Mendel retardation. And this can be excreted to urine also because it's poorly absorbed by kidney. The next is thalassemia. So what is thalassemia? Thalassemia, it is also autosome linked recessive blood, autosome linked recessive. It's a blood disease which is transmitted from parents to offsprings when both the parents are unaffected carrier for the gene. Okay, both parents will be in unaffected condition, but they will be the carriers. Okay, so it's a blood disease which is transmitted from parents to offsprings. Both the parents will be carriers, but they won't be affected. Okay, they are working as carriers, but they won't be affected. And this is autosome linked recessive disease. Autosome linked recessive, in recessive condition, and it is a blood disease. Thalassemia is a blood disease, and in this both the parents are working as carriers, but they are unaffected. And it is, this defect is, or it could be due to either mutation or deletion, which ultimately result in reduced rate of synthesis of one of the globin chains, alpha and beta chain, and make up hemoglobin. Okay, so this causes the formation of abnormal hemoglobin molecules, and that results into anemia, which is characteristics of the disease. What is the special character of this disease? Anemia. So that forms the mutation or deletion of this globin chains. That changes the hemoglobin. Okay, so that change or abnormal hemoglobin, that reduces the absorption, right? That reduces the absorption of oxygen, and also such things. In such condition, anemia, that forms the anemia, that is the main character of the disease thalassemia. Okay, so this is autosomal recessive blood disease, and in this both parents will be working as carriers, but they won't be affected. Okay, it is due to the mutation or deletion of globin chains of hemoglobin. So that changes in the, that make changes in the makeup of hemoglobin, and main character of this disease is anemia. Okay, abnormal hemoglobin may result in the anemic condition. So that is the main character, anemic character, anemia. That is the main character of this disease. Okay. So in thalassemia, that we can classify according to which the hemoglobin molecule is affected. Okay, thalassemia we can classify into alpha thalassemia, because alpha globin chain is affected, means it is called as alpha thalassemia, and it is controlled by two closely linked genes, HBA1 and HBA2. Note down this HBA1 and HBA2. It is alpha thalassemia is controlled by HBA1 and HBA2, and it is present on chromosome 16 of each parent, and it is observed due to mutation or deletion of one or more of the 4 genes. Okay, it is present on 16th chromosome of both parents, and thalassemia, alpha thalassemia is controlled by two closely linked genes, HBA1 and HBA2. Okay, so the more genes affected, the less alpha globin molecules produced. Okay, when it is getting infected more, or more genes are getting affected, or more genes are having this problem, alpha globin chains will be produced in very less quantity. Okay, when beta thalassemia is controlled by single gene HBB on chromosome 11, alpha thalassemia on chromosome 16, beta thalassemia on chromosome 11, 11th chromosome, and it is controlled by single gene HBB gene. It is controlled by single gene. The gene is HBB and it is present on chromosome 11. And each parent, on each parent 11th chromosome, this will be present, and due to the mutation of one or both genes. This may happen due to the mutation of one gene or both genes. Okay, so this thalassemia and sickle cell anemia, both are different. Okay, thalassemia means it is a quantitative problem, but sickle cell anemia, it is a qualitative problem of synthesizing an incorrectly functioning globin. Okay, so there is a difference in thalassemia and sickle cell anemia. Note down the difference also, and you have to learn thalassemia, which is controlled by HBB, and that means you should learn the difference between alpha thalassemia. It is present on which chromosome? It is present on 16th chromosome, while beta thalassemia is present on chromosome number 11. So both difference you need to learn. Okay. So next is chromosomal disorders. So chromosomal disorders, please wait for some more time. Let me complete this chapter. So the chromosomal disorders, it is due to the absence or excess or abnormal arrangement of one or more chromosomes. Okay, the chromosomal disorder means it's a disorder due to the abnormal arrangement or the absence of the chromosomal absence of a particular chromosome, or the excess or abnormal arrangement or one or more presence of one or more chromosomes. So there will be a failure during the cell division and the segregation, in the segregation of the chromosomal segregation during cell division or cell cycle, and that may result in the loss of chromosome, or sometimes it may gain chromosome, or that may, sometimes it is more number of chromosome may come to one side. So in such cases, that is the failure of segregation of these chromatids during cell division. So during cell division, there is a change, or there is a problem or failure in segregating the chromatids. So due to that, these chromosomal disorders are coming. So for example, Down syndrome. Okay, in Down syndrome, extra copy of chromosome 21. There, chromosome number 21, there comes an extra copy. That is the Down syndrome. Next is Turner syndrome. That due to the loss of an X chromosome in human female. Okay, Turner syndrome means loss of an X chromosome in human female. In human female, loss of an X chromosome. That is called as Turner syndrome. Then, failure of cytokinesis after telophase stage of cell division. That results in an increase in a whole set of chromosome in an organism. Okay, cytokinesis means cytoplasmic division. See, after all these cell division procedures are finishing, but final step is cytoplasmic division. It should divide, the cytoplasm should divide, and both cells, daughter cells, should get separated, right? But after telophase stage of cell division, the cytoplasm is not getting divided. That means there is an extra whole set of chromosome, right? Whole set of chromosome is present in an organism, extra set. So that is called as polyploidy. So this condition is seen in plants. Whole set of chromosome as an extra set of chromosome, it will appear, right? Because there is no cytoplasmic division. If cytoplasm is getting nuclear division over, next is cytoplasmic division. So in that step, if the cytoplasmic division is not happening, means the whole set of chromosomes will be present without the whole set. One extra set will be there. So that condition is seen in plants. That is called as polyploidy. Okay. So the total number of chromosomes in a normal human cell, in the case of human cell, it is total number of chromosomes is 46. That means 23 pairs. Total number is 46, but pair-wise when we are calculating, it is 23 pairs. Total number is 46. Okay. So out of these, 22 pairs are autosomes. Out of this 23 pairs, 22 pairs are autosomes and one pair of chromosomes are sex chromosomes, which determines the sex of that human being. Okay. So in human being, total 23 pairs are there. That means 46 chromosomes. Total number of chromosomes 46. When it is asking in pairs, you should give us 23 pairs. In these 23 pairs, 22 pairs are autosomes. Okay. And one pair of chromosomes are sex chromosomes that determines the sex of that human being. And sometimes either an additional copy of chromosome may include in an individual, or they may lack one of any one pair of chromosome. So this situation, this is known as trisomy, trisomy or monosomy of a chromosome. Okay. So such situation that leads to very serious problems or serious issues in the individual. That is called as Down syndrome, Turner syndrome, Klinefelter syndrome, and these are the examples for chromosomal disorders. So chromosomal disorders examples are Down syndrome, Turner syndrome, Klinefelter syndrome, and these are the examples of chromosomal disorders. Okay. In Down syndrome, the cause of genetic disorder is the presence of an additional copy of the chromosome number 21. Okay. Trisomy of 21. Trisomy. Okay. Usually there will be one set, right? Means pairwise. But there is an additional extra set of chromosome number 21. Okay. Additional copy of the chromosome number 21. That is the genetic disorder. Down syndrome, it is a genetic disorder which is caused by the presence of an additional copy of the chromosome number 21. There will be an additional copy of chromosome number 21. So this was first described by Langdon Down. In 1866, they described about Down syndrome, described by Langdon Down. And the affected individual, note down the points, you may get list of symptoms and then you can select which is the disease. Okay, which is that particular disease. Like that you may get 4 options. So don't get confused. Be clear with the concept. So affected individual is short stature with small round head and furrowed tongue and partially open mouth. Okay, so that mouth will be partially open and usually the affected individual will be short stature and they have small round head and furrowed tongue and partially open mouth. And the palm, their palm will be broad with characteristic palm crease. Okay. Then the physical and psychomotor and mental development will be less, or they will be mentally retarded. Physically, mentally, and they will be retarded only. Okay. The psychomotor and mental development will be poor. Okay. This is the Down syndrome. And next is Klinefelter syndrome. In this genetic disorder, this is a genetic disorder and it is caused due to the presence of additional copy of X chromosome resulting into a karyotype of 47. Usually, one human being will be XX in the case of female and XY in the case of male. Okay. So in this, there will be an additional copy of X. So normal number of chromosomes from 46, it will become 47. So in such individuals, overall masculine development and the feminine character, because there is an additional X gene, right? So extra feminine character will be developed. Means development of breast, that gynecomastia, these things are expressed. Okay, so such individuals are sterile. Okay, they won't be fertile. Such individuals are sterile. So with feminine characters, you can see development of breast development and all in such individuals. You can see. Okay. So the condition is X, X, Y. Okay, this is the karyotype. So this is Klinefelter syndrome, and next is Turner syndrome. In Turner syndrome, this is due to the absence of one of the X chromosome. Okay, means 45 chromosomes will be normal. The 46 one, the X determining chromosomes, the X, and there won't be any this another one. Okay, it will be X0. So in such female, they have sterile ovaries. The feminine character won't be complete. Okay, they are sterile and the ovaries. The sterile ovaries and rudimentary beside other features, including lack of other secondary sexual characters. Okay, in those females, the secondary sexual characters also won't be present, or they lack that secondary sexual characters also. Okay, because their condition is 45 plus X0. Okay, so those females will be sterile. You can see short stature and underdeveloped feminine character. Feminine character will be underdeveloped, or there won't be well developed feminine character in the case of such females. Got it? This is Turner's syndrome. So that's all about this chapter, principles of inheritance and variation. Go through that. Okay, is it clear? Do you have any doubt? You want me to repeat any disease or any portion of this? Deepa? Is it clear?
Yes, ma'am.
Okay. So I'll share the questions. Please practice those things also. If you have any doubt, you can ask me in the next class. Thank you.
Thank you.